In the last two videos, we were going over the light reactions, which get their name because they use light to actually start to store light energy so it can be used later on. And then the Kelvin cycle, also called the dark reactions, it's not called this because it happens in the dark. It's called this because it does not use light. So they actually happen pretty much simultaneously in the plant. I mean, the light reactions have to happen first so that you have ATP and NADPH, but they're happening in plants, any time that it's light out. So big picture, the Kelvin cycle uses ATP and NADPH to convert carbon dioxide into sugar. So this is the main idea to take away from this. And if you did the little boxes, maybe they were more confusing than helpful. Maybe you found them helpful. Whichever one you want to do for this. But we'll go over some background and then we'll get into the actual steps. So maybe it's a good idea to hold off on doing any kind of a flow chart till you start seeing some numbers of steps. So this is background on what goes on here. So we're going to use ATP, NADPH, and CO2 in these. You might have noticed that in metabolism, NADH was the reducing or the electron carrier. In plants, there's an extra P here for plant. That's not what it actually stands for, but it'll help you remember it. So it's not NADH, it's NADPH. And it also uses ATP. All right, this Kelvin cycle is going to make a 3-carbon sugar called G3P. Full name is glyceraldehyde 3-phosphate. And then 2 of these 3-carbon sugars end up getting combined to make one 6-carbon glucose molecule. So understand that in biology we kind of go from a series of simple to a series of complex understandings of how living things work. This is no different. You start out by learning that photosynthesis makes glucose. Now you're learning that, well, it actually makes this first and turns that into glucose. All right, there are 3 major steps here. First step is carbon fixation. To fix something in chemistry terms means to affix it, to affix it, so it means to stick it onto something else. So I just fixed carbon onto this N right there. So the first step is fixing the carbon from carbon dioxide onto something else. Step 2 is reduction. Not reduction as in make it smaller, but reduction as in passing on an electron. And then step 3 is the regeneration of this molecule right here, which you'll learn more about later on. But RUBP, or ribulose bisphosphate, this is a really common molecule. Okay, so if you're going the route of boxes, here's step 1 in all of its glory. So step 1, 3 of these carbon dioxide molecules are going to get combined one at a time with RUBP. So here's RUBP right here. And you can see there's 5 carbons, there's phosphates at the end, that's how it gets its name. So ribulose sounds like anytime you see ose, you think carbohydrate, so this is a carbohydrate with 2 phosphates, bi or bis for the 2 phosphates. Don't worry about what this looks like. All you need to know is that there's this stuff called RUBP. It's super common as far as the number of these out there in the world. This is one of the most common molecules in the world, one of the most common organic molecules in the world in terms of weight. So at any rate, here's RUBP. There is an enzyme called Rubisco. It's one of the few enzymes that we'll see that does not have ACE in it. Instead, it's ISCO. So Rubisco fixes this little carbon here onto RUBP. So notice it was not here. There was nothing attached to it. Now it has taken the carbon off of CO2 and it's put it onto RUBP. This says short-lived intermediate. So honestly, guys, being able to read through this diagram is probably worth more than memorizing these steps. So phase 1, it says carbon fixation. You can see the arrows here are showing you that RUBP is coming in here. It's showing you that there's this other stuff called Rubisco right here that takes carbon dioxide, sticks it on there, And a short-lived intermediate means that it happens like this just for a brief second and then it breaks apart. All right, step 2 is going to use, remember the ATP and the NADPH that we made in the light reactions? Well, we use those because you've got to put that energy into building the G3P molecule. So notice in this diagram, here we go right here at the beginning with RUBP. Carbon dioxide enters, so the carbon is adjoined right here. This breaks apart. Now this little guy was the original carbon from the carbon dioxide. ATP comes in here and phosphorylates this. And then we go from this intermediate to this one. Then NADPH comes along, drops off an electron. Anytime you see an exchange like this going on, it's important to note that you're going from ATP to ADP. That means you're putting energy into this reaction. Same thing here, NADPH to NADP+. You're putting energy into this reaction. And then in phase 2 is the reduction. So that is this part right here when the NADPH is dropping off an electron. It is reducing this molecule right here. The output of this is one G3P molecule, and then it regenerates RUBPH, or RUBP. So one molecule, this little carbon entered, bound to this 5-carbon molecule, and now this thing breaks in half. Here is the half that had this little guy on it. The other half you're not seeing in here, it's off in the distance. There's our 3 carbon molecules there. ATP is coming up, it's phosphorylating this, so I'm going to highlight in orange our original. So now that little carbon got fixed, and we still have this guy left over because it's been in the shadows here the whole time, so it's left over. And now we can take our next carbons and we can bind them on here. We can regenerate, use more ATP. We can take other carbons to regenerate RUBP. Okay, so that was the basic gist of this. Spend some time with this diagram. Make sure that you can explain where the carbon is coming from and where the carbon goes. So it's coming from carbon dioxide and it's going into G3P. So this first half of the cycle is really important. What's important to understand right here is that the chloroplast is actually regenerating this initial ingredient. So it's making more of this stuff, which it then uses to carry this through. So the interesting thing about this, if you're thinking about the number of carbons, this is the only new one that we brought in, which means that it's going to take 6 turns of this cycle in order to get 6 new carbon dioxides in through this whole thing. So the first turn through, here's another way of putting this. First turn through, we had one of these joined up on here, and now that has exited right there. Second turn through, we have another one that's joined onto another. Third time through, we've got another one joining on to another. If that doesn't make sense, ask me to explain it another way or go check out some other diagrams. Make sure that you're understanding why it takes 6 whole turns. That's supposed to be a 6 whole turns of this to make it through. So plants that are here in the Midwest in Michigan, they experience a problem. So during a hot day, they can open up their stomata, and as their stomata are open, so there's a view of the stomata from the bottom. Remember, those are the holes in the leaf that let gases through them. So as these stomata are open, oxygen and carbon dioxide are able to get in. And it turns out that while CO2 is pretty good at bonding to rubisco right here, it turns out oxygen can be even better, O2. And so when oxygen binds to this, that is called photorespiration. Remember, respiration, we think in respiration, oxygen is used. Well, this is light respiration. So instead of carbon dioxide coming in and bonding to that, oxygen comes in. Well, what's the issue with that? The issue with that is that when carbon dioxide was coming in here, it was taking carbons and it was using them to build glucose molecules. So we could actually get 6 of these carbons together to make one glucose molecule. When the plant is going through photorespiration, instead, the oxygen comes in there and it's actually going to end up spending way more ATP to make that thing than it will ever get out of there. So it ends up wasting its energy. So it uses oxygen and it produces CO2. It uses up ATP and it produces no sugar. So photorespiration, bad news. This occurs in hot, dry, bright days when the stomata are open. So these stomata, it says when stomata close, this means when they should close. So the stomata, if they close up, there's no issue as far as photorespiration happening. However, you're also not doing photosynthesis. So one hypothesis as to why photorespiration happens here is because in the early atmosphere, remember when photosynthesis first started, there was low oxygen and high carbon dioxide. So it didn't matter if Rubisco was good at joining to oxygen because there just wasn't that much of it around. Well, now it's a much different atmosphere. It's about 20% oxygen. So it's a lot easier for this thing to happen where we're not actually making sugar, but it's instead wasting it.